Greetings to one and all. So this is Balaji from Department of Microbiology, St.Joseph College, Arts and Science, Autonomous Cuddalore. So today we are going to see about bacterial reproductions. So first of all, we are going to see the introduction. So bacteria are the simplest and the smallest and the most successful microorganisms. They were discovered by Anton van Leeuwenhoek in 1976. In the five kingdom classifications, they are placed in the kingdom Monera. So what are the types of reproductions the bacteria may have? So we will see that one is vegetative reproductions, asexual reproductions and sexual reproductions. So vegetative reproductions, it involves budding. So we will see the first budding. In this case, a small protuberance and called develops at one end of the cells. Genome replications follows and one copy of the genome gets into the bud. Then the bud enlarge, eventually become a daughter cells and finally get separated from the parent cells. So this is the thing mainly happens. We will see the image. So for example, I am making this image. So first of all, this is a parent cell. So in first image we can see there is a nothing can present in that image. So while comparing the first to second, you can see there is a something came out from that. You see that? So that is a but from the parent cell it is coming. So it in third one, while comparing to second and third, so it's literally it can grows. You can see the third and fourth, it can partially grows and it can fulfilled. Okay, so in fifth one it can came out from the parental cells and it can came out separately. So in sixth one it can grow. So this is the main process of budding, that is saying that. So in this case, small protuberance. So that is the thing. So in second image you can see that. So small protuberance is producing and called develops at the one end of the cells. So in that end of the cells it can produce, this is called as a protuberance. Okay, protuberance it is presenting. Then genome replications follows and one copy of the genome gets into the bud. So genome replications also occurs and one genome copy will be present came out from that. So that is the in third you can see that. So in fourth it fully developed and fifth from parent cell it can separate as daughter cell. So it's also called as a budding. So in that then the bud enlarge, eventually become a daughter cell and finally get separated from the parent cells. So in fifth image you see that finally it can get separate from a parent cell. So then it enlarges or it grows as a daughter cell. Then we will go to another vegetative reproductions called fragmentations. Mostly during unfavorable conditions, bacterial protoplasm undergo complementarizations on the subsequent fragmentations, forming minute bodies called gonidia. Under favorable conditions, each gonidium grows to a new bacterium. It becomes apparent that prior to fragmentations, the bacterial genome had to undergo to repeated replications on that each fragments get so that each fragment gets a copy of it. So it's nothing but it became apparent to period to fragmentations the bacterial genome had to undergo to repeated replication so that each fragment get a copy of it. So that is nothing better. So this is the parental, like for example I am taking this picture. So it is the parental planaria. So the fragment of planaria, so it can cut it into three pieces and it can grow it, literally you can see that. So that cutted pieces can also grow. This is the daughter planaria. So this is the daughter planaria. So whatever, if we cut into 6 pieces, then it also can grow. This is called as a one type of reproduction. It's called fragmentations. So you can see in that you can so you can see under favorable conditions, each gonidium grows to a new bacterium. It becomes apparent that prior to the fragments, the bacterial genome had to undergo to repeated replications so that each fragment gets a copy of it. So in this, it can cut into 3 pieces, then it grow, and if this 3 can cut it into 6 or 8 pieces, then it can grow up to 64. By multiplication by it's 2 times larger. It can cut into 3 pieces. This one, second one is also cut into 3 pieces, and this third one also cut into 3 pieces. Then we will get 3 3 3. Then it goes to 9, and that 9 again multiplied by 327, like that it can produces. So then binary fissions. So we already know that binary fissions, though it's also one type of vegetative reproduction, as well as it's also called as asexual reproductions. So it's nothing will happen this. That's a one. Simply what we are saying means one cell is present. So in that one cell, it is divides into 2 cells. So first of all, we will see the picture. So we will see it's whether it is a. So this is the picture of binary fissions. So one cell is divided into 2 cells. So this is the main thing happens here. So it is the commonest type of the reproductions under the favorable condition in which cell divide into 2 similar daughter cells during the which is similar daughter cells. So it's a only one parent cell is present. In that one parent cell, it is divided into 2 cells, and another in the 2 cells can be divided into 4. Then 4 is divided into 8, 8 is divided into 16, 16 is divided into 32. Like what it can increasingly it can grow or it can reproduce. During the process, the bacterial chromosomes fits attaches to the cell membrane and replicates to the bacterial chromosomes. All the cells enlarges the daughter chromosomes get separated. A cross wall is formed between the separating daughter chromosomes. It is divided the cell into the daughter cells. So you can note that point. A cross wall is formed between the separating of the daughter chromosomes. So you can see that cross wall. So there is dividing. You can see the dividing. So in that you are seeing the cross wall. So this is the main thing happens here. So this one cell is divided into 2, then its 2 is divided into 4, and 4 is divided into 8. Likewise, this is called as a binary division. So multiplying by 2 times. So for every process, it can grows up to 2 times or reproduce up to 2 times. So that is nothing but cutting and reproducing. So if we cut the cells into 2 parts, means it's also grow as a new cells. So that again I can cut the 2 cells, it can come as 4, like that it will come. So this is called as binary fission. Then endospore formation, we already know that what is endospore formations. So first of all, what is spore? So what is spore means in any unfavorable conditions, the bacteria can produce spore, means it can able to live for long life without changes in any changes in whatever it may be. So in any temperature changes, any climatic changes, or any for example, what we say, if a microbes is there, means it has unfavorable conditions. So in that time it can produces the spore, right? So in that time it can produce the spore. After it is getting favorable, it can release the spore, and it can be normal, and it uh, can be survive. So that is called as spore forming bacteria. So then we will see what is endospore. So endospore or resting spores formed in the some gram positive bacillus and clostridium during unfavorable conditions. They are formed within the cells. So normally spores formed within the cells only. So during this process, a part of protoplasm become concentrated around the chromosomes. So you can see that the part of protoplasm become concentrated around the chromosome only. The hot resistant wall is secreted around it. The rest of the bacterial wall degenerates. Endospore are very resistant to extreme physical conditions and chemicals. So during favorable conditions, the spore wall get reputed and the mass give rise to the new bacteria. So that is the main thing I am saying here. So you see that fifth and sixth point, the rest of the bacterial wall degenerates. Endospores are very resistant to extreme physical and chemical, whatever physical condition at whatever chemical conditions. So it can be extreme resistant. So it's very resistant whatever what physical condition, what it may be high temperature or low temperature. So any chemical it's attack the bacteria. For it's nothing happen to the bacteria. So because there is a formation of endospore, so it can very resistant to all types of chemical as well as the physical conditions. That the thing they are saying, during favorable conditions they can releases the spore, and it's like getting a new life for that bacteria. So this is the picture for the spore formations. So you can see the vegetative cells and medical divisions and can grow germination starters. So you can see stage 6 and 7 maturation cell lysis, stage 5 spore coach, and you can see in spore what are the things present: cortex, cell wall, cell membranes are there. In stage 3 engulfment, stage 2, so it can start it like this. So stage 1 polar divisions, stage 2, stage 3, stage 4, stage 5, stage 6, 7. So it can again germination like that, it can process can happen. So it's conidia. So we will see conidia. Some bacteria such as thermophytes considered as true gram-positive bacteria produces conidia in chain. So like that we can see that. So chain-like structure bacteria we will see, that is called, we will say streptococci. Like that they are saying some bacteria such as streptomyces. Here they are using streptomyces, considered as a true gram-positive bacteria, produces conidia in chain. So I am saying that chains in the arrangement, like a chain shaped arrangement, we know that the bacteria some has a chain shaped arrangement. So that those are all called as gram positive streptococci. They are using here streptomyces because they are saying that produces conidia in chain. Each conidium germinates to give rise to a new bacterial thalamus. So then we will see the sporangiosspores. They are motile platmosphere as are seen in the rhizobium. Then we will move to the sexual reproductions. Sexual reproductions occurs in the form of the genetic recombinations. There are three methods for genetic recombinations. One is transformation, conjugation, and transduction. So first we will see the transformations. So this was first studied by Griffith in 1928 in Diplococcus pneumoniae. In transformation, DNA from the donor cells initiates heritable changes in recipient cells. The transformations involve only one trait, though several may be acquired independently in this way. So just you will see the picture of what is happening. So bacterial transformations. So they are taking any bacteria, so there is a DNA present and also gene also present. By using restrictions enzyme, some of the gene can be cutted and that gene can be inserted into another plasmid of the bacterial cells. So by using ligase enzymes, so ligase enzyme is used for paste that particular DNA. What we are, so restriction enzyme for what purpose we are using means to cut the DNA, whatever the gene, sorry, whatever we want any gene means, we can use that restriction enzyme and we can cut that and we can paste by the ligase enzymes. So it's also ligase means it's also one type of enzyme. You can use it to paste the gene or paste the gene, whatever gene may be we wanted, we can cut it and paste it by ligase. So we can, so at fourth stage you can see gene formed bacterial cells. So it's a new type of gene is formed in that bacterial cells. So this is the main theme of the transformations. So whatever gene we want, whatever gene we have to going to insert, so we can cut those gene by using restriction enzymes and we can insert the gene and paste it by ligase enzyme. Then conjugations. So conjugations, we already know that it can simply we are saying that we can say transformation of any genetic material through pili between two bacterial cells. That is the simply we are saying this thing. So then we'll see detailedly It was the first reported by Ledberg and Tatum in 1964 in E. coli bacteria. Cell-to-cell union occurs between two bacterial cells, and genetic materials of one bacterial cell goes to another cell lengthwise through conjugation tube, which is formed by the sex pili. As a result, the recipient cell possesses duplicate genes for a number of a gene character as seen in E. coli, an inhabitant of human intestine. So normally in human intestines also a lot of E. coli may presence. So you can see the donor F plasmid conjugation pilus and chromosomes. So in one chromosome is present, in one another F plasmid is not present. So these two bacteria can form this conjugation pili. The first image you can see conjugation pili is present. So in that F plasmid two ring na, it can convert it into one ring, it can transfer into the one cell to another cell, and it can stop it, and you can see another in both the cells the F plasmid presence. So it's nothing but a transformation of genetic material from one bacterial cell to another bacterial cell. This is called, by using sex pili, this is called as a conjugation. Then transductions. It was the first of all reported in Salmonella typhimurium by Zinder and Leitberg. In this method, genetic material of one bacterial cells goes to the other bacterial cells by agency of bacteriophages or phages. We can see in virus infecting bacteria. So in this picture we will see, so donor host chromosomes are present in that. A page infects cells here, the donor cell, by normal means. So there is a DNA page present, and the DNA page, so a lot of maybe in that DNA were present. So in that the DNA were inserted. The second image you can see, during replication and assembly of a page particles incorporates a segments of the bacterial DNA mistakes. So this DNA were inserted and it can confuse that, and it can going to mix in that, so it can cut at all the chromosomes. So by that gap, that phage get entered into the cell, and the DNAs can be integrated by that page. You can see the images. So in last and the third image you see, so lot of bacteria phage, lot of pages were get into that cell. So in that chromosomes were taken by that phage. Third one, cell A then lyses the releases the mature pages, including the genetically altered one. Then fourth one, the altered page absorbs to the penetrates another host cell, injecting the DNA from cell A rather than the viral nuclei. So you can see that the fourth image. So after taking that all chromosome or DNA, the cell can releases the phages. Then it can transfer and can sit in another cell. So DNA from the DNA can go to the cells, and the lysis process happening. So in lysis process, that DNA, that gene can transfer that in phage there is a gene, so it that come in the bacteriophage, and it can be join in that new chromosomes and produces a new form of a gene. So by using lysis, so it can break down that particular gene, and it can go and join it. That's the main thing. So when first one, the one phage is going and attaching in a cells, in second image the DNA can get enters, and it can uh, lyses the chromosomes, or separating the chromosomes inside the cells. And third one, the a lot of pages were get inside the cell, and the cutted chromosomes were taken. And fourth one, well developed pages can came out from the cell, and it can again attaches to any other new cells, and then the cells may enter, the chromosome or the gene may enter and produce a lysis process where happen, and it can join with the chromosome and produces a new chromosome. So there's a name called as incorporated into chromosomes. That's the thing happened there. So this is called as a transductions. So these are the topics about the bacterial reproductions. So thank you for sharing the information. So thank you to one and all.